I am Dr. Adeyinka Gbadebo, GM Expression. So, this lecture is a continuation of my previous lecture on mechanism of DNA replication. In my previous lecture, I was able to explain an important component of an important component of DNA replication, and that is the DNA polymerase enzymes. The DNA polymerase enzymes. In this lecture, I also want to elucidate an important component or an important factor that is also needed for DNA replication to begin. For DNA replication to begin, and that is called the primer. Primer. So usually during DNA replication, DNA polymerase 3, POL3, DNA polymerase 3 cannot start the process of DNA replication. It lacks the ability to attach free nucleotides to the unwind end of the parent DNA. It lacks that ability. So usually another enzyme is usually involved, and this enzyme is known as RNA polymerase or primase. RNA polymerase is also known as primase. So this primase is the enzyme that is needed to build a short sequence of nucleotides it's responsible to build a short sequence of nucleotide at the unwind and growing DNA strands on the parent DNA. So the primase built a short sequence of nucleotide, and this short sequence of nucleotide is usually about 10 nucleotides. 10 sequence, usually about 10 nucleotides. And this 10 sequence of nucleotide that is built initially by this enzyme, RNA polymerase or primase, is what is referred to as a primer. So a primer, this is the primer. Primer is built initially. It's usually composed of about 10 sequence of nucleotide, 10 sequence of RNA nucleotides. I think I need to emphasize that. 10 sequence of RNA nucleotides is usually built initially. So it is upon this primer that has been built that DNA polymerase 3 can then start adding free DNA nucleotides to the growing chain of the DNA of the parental DNA that is continually being unwind. So what am I trying to say in summary? The primer is first of all built by RNA polymerase. It's usually a short segment that is built on the unwind end of the template strand. It is this primer that is built that DNA polymerase 3, POL3 recognize and then start attaching free DNA nucleotide towards the replication fork. This is the replication fork. This is the replication fork, and then the chain continues to elongate. After some time, the primer will usually be removed. Will be removed, and the chain elongation will continue. So haven't explained the concept, haven't explained the factor called primer. I also want to explain the I also want to explain what we refer to as leading strand and lagging strand. Leading strand and lagging strand concept in DNA replication. Leading strand and lagging strand in DNA replication. Now let's hypothetically assume this is a DNA double helix. This is a DNA double helix that has unwind at this point. It has unwind at this point. It has unzip at this point. So this point that it has unwind is called the replication. This point. So this point where it unwind is called, or where unwinding starts from, is called the replication fork. It's called the replication fork. That point is called the replication fork. So, and on this DNA double helix, one strand is usually called the leading strand. And the other strand is called the lagging strand. Now, why is it called leading strand and why is it called lagging strand? Now, you need to understand this. In DNA replication, that the double helix, in the double helix, both strands of the double helix are in opposite direction. One strand runs from 5' to 3', while the other strand runs from 3' to 5', and that is what we call antiparallel nature of the double helix. One strand is from 5' to 3' in direction, the other one is from 3' to 5' in direction. So with that, we say the DNA double helix, they are in antiparallel direction. They are antiparallel. The strands of the double helix, they are antiparallel to each other. So if they are antiparallel to each other, in doing replication, for replication to start, remember that we have explained primer. The first thing that takes place is the unwinding. The unwinding at a point called replication origin. This place is the replication origin where unwinding takes place. And unwinding is aided by an enzyme called DNA helicase. Unwinding is aided by an enzyme called helicase. DNA helicase aids unwinding of the double helix. So when unwinding starts, it will create what we call a replication fork. Unwinding creates a replication fork. Unwinding takes place on a short sequence of nucleotide called replication origin. Unwinding takes place on a short sequence of nucleotide called replication origin. DNA polymerase 3, the enzyme that is involved in DNA polymerase, can only attach nucleotides, free nucleotides. DNA polymerase 3 can only attach free nucleotides in the 3' to 5' direction on a growing DNA strand. Can only attach free nucleotides to a new strand on a growing strand following this direction, 3' to 5' direction. Note that also DNA polymerase on the parent strand, DNA replication on the parent strand usually and can only take place in the 5' to 3' direction. I would like to repeat that DNA polymerase 3 can only synthesize the new and growing strand in the 3' to 5' direction. However, DNA synthesis of the parent strand can only take place in the 5 prime to 3 prime direction. So having said this, because DNA synthesis, DNA replication can only take place in the 5 prime to 3 prime direction. This tells us that the direction in which synthesis will be taking place on this strand will be different from the direction in which it will be taking place on this strand. While synthesis of the new strand for this strand will be taking place in this direction. Synthesis by DNA polymerase 3 will be taking place on this strand, while synthesis of DNA by DNA polymerase 3 on this strand will be taking place in this direction. That is from 5 prime to 3 prime direction. Will be taking place in this direction, 5 prime to 3 prime direction. However, on the other strand, it will be taking place in this direction, which is also 5 prime, it will be taking place in this direction, which is also 5 prime to 3 prime direction. So once DNA synthesis or DNA replication is about to commence, like I've explained earlier, the first thing that takes place is the building or construction of primer. So let's assume this is a short primer that is constructed by the primase, by RNA polymerase known as primase. It's first of all constructed, and then once this is constructed, DNA polymerase 3, pol 3, then recognize this primase, this primer, and then begins to build nucleotides to elongate the primer, along the parent DNA. Now, I want to note that DNA synthesis, this DNA, to synthesize this DNA, to replicate this DNA, it can only take place in the 5 prime to 3 prime direction. It can only take place in the 5 prime to 3 prime direction, 5 prime to 3 prime direction. So DNA synthesis on this strand can only take place in this direction, and then on this strand it can take place in this direction. Now, that is for DNA synthesis. On this strand it can only take place in the 5 prime to 3 prime direction. And then polymerase 3 builds the new strand in the 3 prime to 5 prime direction. 3 prime to 5 prime direction. 3 prime to 5 prime direction. Towards the replication fork. Builds in the 3 prime to 5 prime direction towards the replication fork continuously, and that is being built continuously on the leading strand. DNA polymerase 3 built the newly synthesized DNA strand continuously on the leading strand towards the replication fork. However, on the lagging strand, that is why it is called lagging strand, since this synthesis can only take place from the 5 prime to 3 prime direction. Primer is also constructed. The primer is constructed on the open end. In this way, and primer is constructed by RNA polymerase, and DNA polymerase III also builds sequence of DNA nucleotides on the primer in this direction, away from the replication fork, away from the replication fork, away from the replication fork, and that will, that will lead to synthesis on the lagging strand to be in short segments. That will make synthesis of DNA on the lagging strand to be in short segments. This short segment is what we call Okazaki fragments. This short segment is what we call Okazaki fragments. Usually Okazaki fragments in eukaryotes, they are usually about 100 to 200 nucleotide long, 100 to 200 nucleotide in length. In prokaryotes, they can be up to 1000 to 2000 nucleotides in length, nucleotide sequences in length. So the Okazaki fragments, they are usually short segments which are built on the lagging DNA strand, and eventually these Okazaki fragments, they are joined. Short fragments are, this DNA will continue to unwind, and then short fragments will be synthesized, and these short fragments called Okazaki fragments will eventually be joined together by DNA ligase. It will be joined together by another enzyme called DNA ligase. So this phenomenon where DNA polymerase or DNA synthesis on the leading strand is different from the DNA synthesis on the lagging strand. It's different from the way it is done on the lagging strand. On the leading strand, it is synthesized continuously. On the lagging strand, it is synthesized discontinuously. This phenomenon makes DNA replication process to be referred to as a semi-discontinuous replication. So DNA replication follows a semi-discontinuous process. DNA replication follows a semi-discontinuous process.